Hello dear students. I was discussing about acetylation of core histones. The core histones consisting of H2A, H2B and H3H4 tetrameric structures. Together it forms an octamer. So here the acetylation is going to happen at the lysine amino acid. So which I told you in the last class, the the lysine becomes N-acetyl lysine, epsilon N-acetyl lysine, and this particular acetylation is going to happen with the help of histone acetylase. So this particular activity of acetylation is going to change the nature of the side chains or the nature of the histone chains, because histones at the N-terminal region, it's going to have a series of amino acids having positively charged. Specifically they are going to have these lysine amino acids, so which are going to interact with the DNA and holds it very tightly. Now because of this acetylation, you can see they are initially positively charged. So now because of this acetylation, what will happen is that those particular positively charged histones are now going to become, you can see the positively charged histones are going to become um, neutral. So because of this neutralization of the charge on the lysine amino acids are the histone chains, so the interactions between the DNA and that particular histone is going to get loosened up, so that this particular DNA now it is going to become partially available for transcription, or it is going to get loosely interacted with the core histone molecules, so which is going to help in further it can involve in the process of nucleosome repositioning or positioning. So histone acetylation has been linked with the transcription. People have done the experiments wherein they have identified that when the acetylated histone tails are going to be there, so then transcription is going to get increased. So which people have actually confirmed by using different experiments that the chromatin immunoprecipitation experiment, and also people have done the mutations of the neutralizing mutations on the histone tails, and they have checked this neutralization wherein they have removed this acetylating amino acids from the histone, that can lead to decrease in the transcription rate. So acetylation is going to make the nucleosomal DNA more accessible for transcription factors, which can include, which has a role in the potentiation or the initiation of the transcription. So you can see these are the different core histone proteins, and these core histone proteins are going to have different number of these lysine amino acids within the N-terminal chain. So you can see K is the lysine amino acid which undergoes acetylation, and which is going to change the nature of these lysine amino acids. So histone tails not just only undergo acetylation, you can see they undergo multiple type of modifications. For example, the arginine lysine, even they can undergo the modification by methylation and acetylation, as well as the serine amino acid can undergo phosphorylation. Okay, so these are the few type of modifications: acetylation, methylation, phosphorylation are the few type of amino acid alterations which are going to happen, or modifications which we'll see in case of histones. Okay, so this UB is nothing but ubiquitination. So now what is this particular enzyme is going to do, how it's going to look like. So I explained to you earlier, like GNZ5 is found to be the one of the most well studied enzyme of the histone acetyl transferase family, or histone acetylases. So wherein this particular hat histone acetyl transferase GNZ5, you can see it's how it looks here with the ribbon model, and what it does is lysine conversion into the uh, epsilon N-acetyl histone. So this particular enzyme is going to catalyze the transfer of acetyl group from the acetyl CoA to the histone tiles, and there are two different types of histone acetylases, that is hat A, hat B. So whereas hat A is going to be found to related to the transcriptional acetylation, whereas hat B is found to be playing a role in the cytoplasmic role and catalyzes the acetylation of newly synthesized histones, okay, from cytoplasm to the nucleus. So along with this acetylases, we also come across with enzymes called deacetylases, which are called as HDAC, histone deacetylases. So what these HDACs are going to do, they convert the acetylated amino acid into the normal amino acid. They remove the acetate moiety or acetyl moiety from the amino acid. So there are different hat families are there. You can see GNAT family, which includes GNC5 related type of histone acetyl transferases, and they contain bromodomains, many bromodomains, wherein the changes are going to be seen. So you can see East GNC5 and H PCAF, and there is the MYST family that is named after the founders of this. They called as MYST family. There is MOS, YBF 2, SAS 3, SAS 2, TIP 6, like that. These are the few histone acetyl transferases of this MYST family, and they contain several chromodomains in that. So just remember this chromo and bromodomain wordings. So I'll explain about what is this bromodomain, what is the role of it, what is chromodomain, what is the role of it, and how they play with a role in the modifications of chromatin. So another is CBP slash 3 P300, and there is GTF hats. TAF2 250 is an example of GTF half, general transcription factor histone acetylase families. The nuclear receptor linked hats are also there, SRC1, ACTRs. So these are also going to play an important role in the acetylation of histones. So few of these particular are going to be involved in the process of regulating the transcription. Few of them are going to be involved in the regulation of they call other aspects like movement of histones within the from the cytoplasm to the nucleus and etc. So this is an clear cut complex of hat 3 with the H3, hat with the H3. So the structure of hat domain, so particularly in case of GNAT family, we can see there is a central conserved core unit which is important for the acetyl group binding, the acetyl CoA binding, and a cleft within this particular structure. You can see the cofactor or the acetyl moiety is going to bind here, okay. So sorry, and then there is a cleft which is used for substrate recognition that lies directly over the cofactor pocket. So this is the cofactor pocket which you can see there, and directly over that there is substrate binding pocket. So whereas oppositely you can see the blue color dots, the ball and stick model of H3 which is binding with the ribbon. The ribbon model is the hat, okay. So this is a pinkish one is the cofactor, the acetyl CoA. So that they are going to be directly interacted within this particular enzyme pocket. And the peptide binding pocket, the H3 peptide bound to the GNAT GNC5 hat adopts a random coil structure and extends the backbone. The extended backbone uh, 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 contacts with this particular coil structure. You can see there in the blue one, the H3 the this is the H3 tail actually, which interacts with the hat. And the phosphorylation of serine 10 in case of H3 generates additional interactions with the hat GNC5, and it also explains how actually histone modifications are going to influence one modification on the other modifications. So which is relatively like if there is a serine 10 phosphorylation, then it increases the phosphorylation acetylation at lysine 14 means. So this is like one modification influencing on the other modification in by increasing the chance of modification. So there is sometimes unexpected large number of hat complexes are also seen. You can see several co-activators have like a hat-like activities, and there are evidences for genes specific hats are also there. Okay, so these are examples you can see. So HTAF 250, 230, 130, and there is CBP P300 complex, GNC5, like that. They are the co-activators, whereas gene specific hats like you can see P300, CBP, PCAF, or GNC5, like that, or CBP hats. There are many number of hats which are going to be playing an important role in the process of activation of a particular gene or transcription of the particular gene. You can see how the hats are influencing the operator, promoter, sorry, and enhancer regions and all. So histone modification, it's a highly specific process, so it's not like everything is going to modify everything. Now you can see there, like these particular histones, sometimes they are going to be freely can involve in the modification, sometimes they are going to be part of another complex and involves the modification. So example, you can see GNC5, it can alone also involves in the modification, where H3 is going to get modified at its side chain, and also its saga, it can involve in the modification of the side chain. There is new A4 and ESCF1 complex also involves in the process of modification of H4 as well as H2A. So activation or silencing of a particular gene is also is decided between, like people say, hat and HTAC, because the equilibrium between these two in terms of their activity and in terms of the histones acetylation, so that decides means how the means whether a particular gene is going to be more accessible and more active in terms of transcription, or less accessible and average transcription it undergoes, like that. So acetylation is linked with the chromatin activation in terms of transcription. So there are many activators which acts like a hat, and even a hat is also going to be there. And even histyle acetylation is found to be very much directed phenomenon, and present model says that hat activity becomes recruited to promoters through a specific interactions between the activators, and this particular complex results in the acetylation of nucleosome around that particular promoter, and makes the transcription possible. There is global versus targeted acetylation and deacetylation. There is a global acetylation that is called bulk acetylation, whereas many number of core are going to get acetylated. So it need not to be at a particular gene or a particular nucleosomes. The acetylation is going to happen over 13 to 30 tiles of tiles lysine residues are going to get acetylated, and this acetylation is going to be maintained by opposing actions of hat and H-TAC complexes together. So targeted, in this particular case, the hat and HTAC complexes, they are going to complex with the promoter region, and creates a specific pattern of hyper and hypoacetylated the core histones, so which specifically involves in the regulation of transcription of the specific genes. So hat and coactivators, they sometimes can be in the same complex, and causes the acetylation, you can see. So US region combined with the act1, and then the complex of ADA complex having GNC5 hat involves in the acetylation of the histones carrying the promoter region. There is saga complex which also involves in the means activation. So here is the coactivator bridges. Sometimes coactivators are going to act as bridge and helps in the acetylation. So you can see ADA and SPT complex and Saga SPT complex, they're all going to involve in the process of acetylation and then uh, causing the activation of the gene in terms of transcription. Now moving to what is this bromodomine, chromodomine, we'll see. These bromodomines, they are the binding modules for acetylated histones. Bromodamines recognizes the acetylated lysines on the histone tails. So means the proteins carrying the bromodamine structure, they specifically can go and bind with these acetylated lysines, and this particular domain is going to be consisting of about 100 amino acids. It's a motif which is going to be seen in many number of proteins which has a capacity of interacting with the DNA or which are going to be involved in the process of modifying the nucleosomal structures, including the hats. So 4 helix bundle with the hydrophobic pocket able to interact with the acetylated lysine, that is the how actually bromodomine is going to be. This is the um, bromodomine of H4 complex which is going to interact with. So bromo in case of hat, it is going to be plays important role in recruitment of other bromodomine containing transcription factor. Sometimes it will have these double bromodomines. Okay, so going to the core histones and on the other side interact with the one side promoter is interacted, the other side the histones are going to be interacted. Other histone modifications, like you can see, there is phosphorylation, methylation, sorry, and phosphorylation, like serine 10 undergoes phosphorylation, happens with the EGF stimuli. RSK2 mediated phosphorylation is going to happen. Is methylation, histone methyl transferases are there, HMTs we call them as. Is ubiquitinylation. So in case if the particular histone is completed its half-life period, it's supposed to undergo the uh, process of degradation, so wherein the ubiquitination is required. So histones are the highly conserved proteins in terms of evolution, and also uh, among the most variable in post transcriptional modifications. Now, so I just explained how the acetylation is going to be there and how it affects the process of transcription. Now into the methylation. So this methylation is found to be having a role in both transcription and repression. So methylation is a very important process which even you can see it will help in the identification of parental and daughter strands during the mutational repair. So if you remember, which we discussed earlier. So this is what the methylation is. So there are several type of methylation, which several rounds of methylation also can happen at the histone tail. So lysine can undergo monomethylation in single methylation, double methylation, triple methylation, leading to formation of monomethylysine, dimethylysine, trimethylysine, like that. So you can see the structure of dimethyl, trimethyl. So they can be symmetric, asymmetric. All these kind of natures are going to be established, and both even arginine also has a capacity of undergoing methylation. So this methylation is a kind of relatively more stable modifications which also protect the DNA from the host DNases are the restriction enzymes. So it has a very slow turnover rate, and that means demethylases are means not much found. And an ideal epigenetic marker is this methylation axis, and methylated residues are present in both U and heterochromatin regions. So if you remember, U is active part, hetero is inactive part, like that. So the histone methylation, the substrate for this methyl transferases HMTs is the arginine and lysine. You can see how exactly the reaction is. Arginine, how it get transferred with the methylation. PMRT is the enzyme. how it transfers the methyl groups, resulting in the formation of symmetric asymmetric one. There is type 1, type 2 PMRTs, there is HMT. Okay, and you can see that basicity of the lysine increases with the increase in the methylation. Histone tiles are going to get multiply methylated. You can see how they means in case of H3 more methylation is possible, H4 plus, mm, like that. And the color coding you can see there, just mentioned. So methylation at the top, if it is going to happen in the methylation, if it is going to happen like 79 36 4 in case of H3 induces activation of the gene, whereas methylation of K9, that is K9 as well as K27, can cause repression, because I told you earlier methylation has both repressive activity as well as what do you call it as transcription activator activity also. So histone lysine methyl transferase HKMT is, we can call it as the first HKMT which was identified was SUV39H1. It was identified in year 2000, and it is specifically methylates lysine 9 of histone 3. So it has a domain called ACT domain. It is required for the HKMT activities. So that is methyl transferase activity. 5 lysines within H3 and 1 lysine at within H4 are going to get methylated by these HKMTs. You can see the table. It is mentioned about the catalytic subunit of the enzymes in different organisms, how they are going to be involved in the methylation. So if you come to the important domain, the ACT domain, so this ACT domain is found to be highly conserved domain in case of HKMTs, and in case of humans there are more than 30 ACT domain proteins have been identified in yeast 6, and altogether there are more than 300 has been found. So you can see this is the structure of DIM5, a homolog of SUV39H1 protein. So it methylizes the lysine 9. You can see how it is interacting with acetylnosyl methionine is a cofactor which transfers the methyl group for the methylation, and you can see the color coding of different components. There will be zinc, okay, and there is orange color is going to be for lysine, which you can see there H31, and there is H3 tile of in the blue color, and cofactor is in purple, and zinc in the zinc ions are there, which is going to be involved in the altogether causes the methylation. So this methylation, as I mentioned earlier, it's not just only involved in the process of repression or transcription, it also being considered as one of the important epigenetic marker and involves in the long-term epigenetic maintenance. So there is heritable changes in the gene expression, so that do not result from the alterations in the DNA sequences, the epigenetics. So here the HMTs involved in this particular process for short term as well as long term in case of regulation of transcription. So it has both activation and repression. There are two sites, two HMTs, there will be two effects. You can see in case of H3, this is H3. So this is R K9 and 4 is their position which can undergo the methylation. So K9 when methylates, it is found to be more methylation in case of heterochromatin formation. K9 undergoes methylation, whereas K4 methylation is found to be related to the transcriptional activation. So this in case of H3, people have done this methylation studies very much well. So this is how the people have found. So K9 methylation causes the particular portion of the nucleosomes or the chromosome to become heterochromatin structures where transcription will not be seen, and if K4 is going to get methylated, so then transcriptional activation is seen. So set1p associates with the hat complex causes methylation of K4, and SUV39H1 causes K9 causes repression. So that's what here repression and activation both is going to happen. So numerous reports is there regarding H3K9 methylation and conversion to heterochromatin. So highly condensed centromeric regions is related to methylation at K9 and H3K9, and H327 methylation is also found in case of X chromosome in activation process along with K9. In activation, H3K4 is found to be more involved, even H336, K36, K79 also involves in the activation of gene and converting the heterochromatin into euchromatin region and preventing to become heterochromatin region. So activation effects of methylation is going to be at two levels. At chromosomal level, preventing the spread of heterochromatin means conversion of euchromatin into heterochromatin. Thereby it is going to make the particular part of DNA available for the transcription. So here methylation of H3K4 specifically impedes the methylation at H3K9, thereby blocks the conversion into heterochromatin, and binding of deacetylases like NUR3, NURD. So repression complex for H3 N-terminal tail. So it also precludes the methylation at K4 and converts into K9, means induces K9 methylation and heterochromatin formation. So at gene level regulation, or the role of this methylation is, so dimethyl H3K4 appears to be global epigenetic marker for euchromatin region. Trimethylation of is found to be more correlated with the active transport and more elongation later. So you can see so how NERD is going to block K4 methylation, induces K9 methylation, converts into heterochromatin. On the other hand, set 9 is going to methylate K4 and avoids K9 methylation. So there is dot 1L found to be methylating K79, and this K9 methylation is going to cause heterochromatin. K4 methylation converts into euchromatin. Now the second type of domain which I was supposed to discuss is chromodomain, so which again plays important role in the process of modification of chromosomes. So methylated lysines are recognized by a conserved domainal structure in different proteins called a chromodomain. Okay, so the chromodomain binds to the histone tails bearing the methyl lysine in highly specific manner, and affinity is found to be very highest with the trimethyl lysine and lowest with the monomethyl lysine. So this is the chromodomain protein binding. So you can see the H3 with the methylation is going to interact with the this chromodomainal binding protein having helix as well as sheets together. So this recognition of methylated histones happens with the specific chromodomain proteins. The number of chromodomain proteins are there. HP1 is one which binds with the methylated histones specifically by SUV39H1, where methylation is going to happen at H3K9, so converts into heterochromatin. So formation of heterochromatin happens with the protein, that is HP1, and this protein binds to this methylated H3K9 and converts into means it propagates the methylation of the other nucleosome at the H3K9 position, converts them into euchromatin region. You can see the same thing. So K9 methylated by the SUV39H1 HMTs will bind by HP1s, and multiply it's going to help in the methylation of other H3K9s, converts a particular chromosomal structure into an heterochromatin structure. So that's how chromodomines plays important role. So people have found this H3K9 methylation is a molecular switch between U and heterochromatin region. So how this molecular switch acts, you can see. So methylation at H3K9 converts a particular DNA portion into an heterochromatin structure where it is going to become non-available for transcription. You can see chromatin by the HAT kinases and HDK phosphor protein phosphatases. It can become normal nucleosome, which can further get converted into an heterochromatin structure by the SUV39H1 methylation of H3K9, convert it into an heterochromatin structure. So further, this is arginine methyl transferase, HRMTs or PRMTs. The protein arginine methyl transferase also they called as. So several HRMTs or PRMTs are identified, and the CARM1 and 1 coactivator associated arginine methyl transferase. So you can see there are several of these proteins, so which are involved in the process of transferring of methyl groups to the arginine. So this is the PRMT domain, which specifically involves in the transfer of methyl group. So here donor will be the SAM in methylation, acetylinosyl methionine, which transfers to the guanidino group of the arginine to produce monoarginyl monomethyl arginine or dimethyl arginine. Type 1, type 2, if you remember, we discussed earlier. Sorry. So what's the function of this methylation of arginine? Specifically, methylation of arginine at H3 and H4 correlates with the correlates with the transcriptional activation, and that is methylation of H3 at arginine 3 at H4 facilitate H4 acetylation and enhances the transcriptional activation. And removing of methylation happens by means it's very rarely seen process, because methylation is the most stable modification and remains for a long time. It is considered to be a kind of irreversible modification, and histone demethylases have not been identified till now. So still there is possible to remove. A process exists that removes the stable histone methylation, so that can happen through a exchange of histones, modified histones. There are some other kind of modifications which can see PRP, the addition, that is PRP recruitment. So during the activation of a particular gene in the on PRP is recruited by the transcription factors onto the condensed chromatin structures, you can see there. And so modification is polyadenosyl ribosylation, so ADP ribosylation. So PRP induces dissociation of nucleosomes and thereby makes the DNA available for transcription, and it stays open. That is, it prevents the rebinding of transcription factors to the DNA by adding ADP ribosyl to the tails. That is, histones are going to be kept separate so that transcription can easily takes place. There is one more concept, a very important concept we come across, is called histone code. So this histone code part, I think I'll discuss in the next video. So just remember, these are several varieties of modifications are going to take place at the histones, starting from methylation, phosphorylation, acetylation. So these modifications are going to be well read by a specific type of proteins carrying either bromodomain for acetylated lysine, chromodomain for methylated lysines. So and this methylation has a very important role in conversion of means euchromatin protein into an euchromatin region into an heterochromatin region, and it also plays as an epigenetic marker, the methylation. So thank you. So take care, keep washing your hands.